Alright guys, I'm going to do a quick little video about genetic engineering because I know that some people are a little confused about it. So if you take a look here, I have a human cell, and here's the cell membrane, here's the nucleus. Inside the nucleus I have DNA. This DNA is in loose form, so it's called chromatin. Now, I drew this here to illustrate what the DNA could look like if I was to stretch it out. And inside your DNA, you have sections that are not used to create proteins called introns. And then you have different sections representing different genes. So it could be a gene for hemoglobin, a gene for insulin, a gene for antibody, whatever it might be, but different genes for different proteins. Now here we're going to look specifically at insulin because people with diabetes have difficulties making the protein insulin and therefore have difficulties taking the glucose from their blood into their body cells. So they need to get insulin in some form. So for us to do this, we don't want the gene A or gene C. I just want gene B to be able to be removed. So as we remember from genetic engineering and also from gel electrophoresis, if I use enzymes, I can cut DNA. Now there are very special types of enzymes called restriction enzymes. And restriction enzymes, similar to that state lab that you guys did, cut DNA in a very specific sequence. Now here, this restriction enzyme is going to recognize the sequence AA, TT, and make a cut. So it makes a cut right here. If you also notice, this says AATT from the opposite direction, so it's going to make a cut, and it kind of makes a zigzag cut, forming these things called sticky ends. Now these TTAA don't like being by themselves. They like to be with their complementary pair. Now this is going to come into play when we talk about creating the recombinant DNA. Now once we have our insulin removed, we want to insert that insulin into a bacterial cell because bacterial cells reproduce rapidly through asexual reproduction. Bacterial cells have their DNA in the form of plasmids, and if I remove that plasmid and I open it up using restriction enzymes, I'm going to be able to insert that insulin gene inside. So now doing this, I need to use the same restriction enzyme. So they recognize that AATT, AATT, forming that same zigzag cut and the same sticky end. So as you notice, you're probably thinking AATT is going to want to go and stick with the TTAA of that gene, and this one's going to stick with AATT from the human. And that's going to create, our recombinant DNA. So as you can see, bacterial DNA, human gene, insulin gene, the complementary pairs match up, and then adding a little ligase, which is an enzyme kind of used like tape, will help to bond the phosphates and sugars together, forming a nice recombinant ring. So I put that human insulin gene inside the bacterial DNA. Now this is just DNA. DNA is used to create proteins. However, we need ribosomes to create proteins. We need RNA to create proteins, amino acids to create proteins. So there's a lot of machinery and ingredients required for protein synthesis, and it's just not sufficient just having the DNA. This is kind of like me saying, here, I have a cookbook, and this cookbook, how can I make my food that I want to make without a kitchen, without ingredients? It's not useful until you put it inside a cell. So here I take this recombinant DNA and I insert it into an empty cell, and now I have a transgenic organism. Transgenic because it's genetic information from multiple organisms or two different organisms. And here I have the recombinant DNA. Now going back to that cookbook scenario, which I'm going to keep going back to, here's your cookbook, your normal cookbook. And I said, hey, do you want my zucchini bread recipe? And you're like, yeah, that's awesome. Let's definitely make zucchini bread. Can I definitely have your recipe? So I give you my recipe and you put it part of your cookbook. That's like recombinant DNA. My zucchini bread recipe is this inserted donor gene, and the blue part, that plasmid, is like your normal cookbook with all your recipes. Now this cell, when it has that recombinant DNA, doesn't realize it's different. It's going to go on its normal business, go through asexual reproduction, producing lots of identical cells from that one, and now I have many, many cells that all have that recombinant DNA. Now, since these cells all have that insulin gene, they're going to be capable of making insulin. Now, how many of you go into your cookbook every single time and make the same exact thing? I'm sure many of you are saying, not me, or some of you are saying, I don't even know what a cookbook is. Well, in my case, I don't go into the cookbook and take out the same recipe every single time. It depends on the environment. It depends on what I'm making. If I'm making zucchini bread, brownies, banana bread, cupcakes, whatever the case is. So this cell has the ingredients, the instructions to make insulin. However, the environment has to tell it to do that. So this cell makes insulin. This cell makes insulin. This cell makes insulin, but if you notice here, this cell didn't make insulin. It has the instructions, but the environment say, no, turn off that gene and don't make it. So this is where gene expression plays a role. Even though the gene is there, the gene must be expressed in order for proteins to be produced, and gene expression doesn't always happen. Again, this is genetic engineering. It's a technique that we can use. It's not just used with bacterial cells. I can use this to insert a desirable gene, such as a gene that insects don't like and will keep them away from plants, and I can insert that gene into plants. Or I can insert an antifreeze gene into plants. So I could take any 2-3 organisms' genes, insert them together, and now I have genetic engineering. Hope that helped to explain it.